Hi to all of you. In this bioconcept lecture, we'll be discussing ribosome structure, a little introduction about the ribosomes and what their structure are, and then the comparison between the prokaryotic and eukaryotic ribosomes will be covering in this lecture. So before we begin and tell you about the ribosome structure and function, the importance of ribosome can be realized that the elucidation of its structure and function has won two different Nobel Prizes. One was in physiology and medicine in 1974, which was awarded to three different persons, sharing it in 1974 for their discovery. And then recently in 2009, Nobel Prize in Chemistry was awarded to three different persons as well for the detailed structure and mechanism of the ribosome factory. So this is the importance of the ribosomes possess in the cell structure and function. Ribosomes in its chemical structure are actually formed of proteins together with the special RNA, which is called as rRNA, R for the ribosomes. They make up the most stable forms of the RNAs out of the 11 known forms of the RNAs in nature. They are the actual seeds where the translation process takes place. The decoding of mRNA, which happens in the ribosomes, are from the 5' end, and the ribosomes are actually the seeds where the peptide bonds are formed between the two amino acids with the help of their own intrinsic peptidyl transferase activity. Ribosomes in both prokaryotes as well as eukaryotes have two subunits, large and small. Small subunit of the ribosome is the one which is the first to interact with the decoding mRNA and able to read and bind to it first. Then followed is the large subunit, which possesses the actual catalytic activity of peptidyl transferase, binding the two amino acids together by forming peptide bond. This peptidyl transferase is located in the special site, which I will cover later. And this large subunit is almost always associated and bound to amino-acylated tRNA. Prokaryotic ribosomes are referred to as 70S ribosomes. They are about 2.7 into 10 raised power 6 molecular weight, and they have two different subunits. The large subunit is 50S and the small subunit is 30S. And large subunit is made up of two different types of RNAs, which is 5S and 23S. Together with the 36 protein, they make up large subunit. And the small subunit is of only one RNA type, which is 16S, and is associated with 21 different kinds of proteins to make the structure 30S. Together, when 30S and 50S come together during the translation process, they make up 70S, complete ribosome to translate the proteins. And this is the composition which you can see in the tabulate form also, and their respective weights. Usually these ribosomes are synthesized from the nucleoid in the prokaryotes by RNA polymerase, which already is present in the prokaryotic cell as only one form of polymerase, which is able to decode the DNA into the RNA. And since there is no differentiation in the prokaryotic or bacterial cell, everything is present in the cytosol. The ribosomes are also present in the cytosol to do their translation function. Eukaryotic ribosomes are little complex and having a high molecular weight than that of prokaryotic ribosomes. Here they are called as 80S ribosomes, and they have a molecular weight of 4.2 into 10 raised power 6. Again, as in prokaryotes, they are made up of two different subunits, large and small. Large subunit is called as 60S, and the small subunit is called as 40S. 60S large subunit is made up of three different kinds of rRNAs, which are 5S, 5.8S, and 28S rRNA. And together with the 49 different kinds of proteins, they make up 60S large subunit. While as 40S small subunit is made up of only one of the types of rRNA, which is 18S. and this gets associated with 33 different kinds of proteins to make up small subunit. Together, both of them make a submolecular assembly called as the ATS, eukaryotic ribosomes. Further, you can see the comparisons of the composition of eukaryotic ribosome in the associated table as well. Also, since we know that DNA synthesizes the RNA via RNA polymerases, here polymerase 1 is responsible for the synthesis of a 5.8S, 18S, and 28S rRNA from the polycistronic 40S DNA modulatory repetitive type of a DNA, which I have covered in COT analysis in the molecular biology classes. You can see how it gets synthesized from the 45S polycistronic gene. However, the DNA polymerase specific for the synthesis of the rRNA here is Pol1. However, the fourth type, which is 5S rRNA, is synthesized from the DNA by Pol3. Also, since there is a differentiation in the eukaryotic cell and every kind of an organelle is involved and dedicated for different kinds of functions, ribosomes here most of the time get associated itself with the endoplasmic reticulum together with the protein present in the endoplasmic reticulum called as riboforins. Riboforins get associate themselves with the ribosomes and result in the formation of a rough endoplasmic reticulum. So in eukaryotic cells, they are present on the surface of a endoplasmic reticulum where the synthesis and the translation process takes place. Last but not the least, what are the different sites of the ribosomes? In prokaryotes, ribosomes have been found to have three different sites. They are called as the A site, P site, and E site. A site is called as amino acyl site, which accepts the amino acyl tRNA, charged tRNAs, during the elongation phase of the translation. While as the P site is the middle site, which is the actual site where the peptide bonds are formed between the amino acids. This site possesses the peptidyl transferase activity. And then the last site is called as exit site, which is the site where the uncharged tRNAs leave the ribosomes during translation process. However, this E site is present only in prokaryotic ribosomes. In eukaryotes, only two sites are present, A site and P site. If you like the contents of this video, do visit my Amazon page for the molecular biology and biotechniques book from which I have made the lecture. You can order it in both Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.